Good morning, everybody. Can you guys hear me okay? Excellent. Welcome back to campus. It's really great to have you here in person. I'm Mindy Rex. I am on the College Relations and Development team here in the College of Chemistry. And I am pleased to introduce our speaker today, Alanna Schepartz. Alanna is the TZ Chu and Irmgard Chu Distinguished Chair and Professor in the Departments of Chemistry and Molecular and Cell Biology here at Berkeley. Alanna came to us from Yale in 2019, where she was the first woman to be granted tenure in Yale's Department of Chemistry and the first female full professor in any physical sciences department at Yale. So we're obviously very happy that she decided to come to Berkeley, and she just told me that she's happy to be here too. Alanna and her research group are known for the creative application of chemical principles to understand and control biological recognition and function. Her research has contributed to and shaped thinking in multiple areas, including the mechanisms of protein DNA recognition and transcriptional activation, protein design and engineering and their application to synthetic biology, and the complex process by which chemical information is communicated across biological membranes. All of that, you may know what that means, and we'll learn a little bit more about that. So Alanna is a member of the National Academy of Sciences, and she's received so many awards that I just am not going to list all of them because we would never get to her talk. In her spare time, Alanna likes to spend time with her family and their two very large dogs, where she likes to hike the canyons here in Berkeley. And despite Alanna's long list of accomplishments, her biggest claim to fame, stated by her, of course, other than coming to Berkeley, is that she won the Wonderama dance contest in 7th grade on TV. Yeah, I know, that's notable. Yeah. So I realize it's probably too much to ask her to reenact the dance, but I know that she's delighted to be our homecoming speaker today. So please, let's welcome Alanna Schepartz.
So thank you very much, Mindy. It is really true. I won the dance contest, and one of the prizes was a purple banana seat bicycle. And I just have to say, so it was a crazy thing. So we had, my sister and I accompanied Dory and Elise Kimmelman, whose parents somehow procured the tickets, which were very difficult to come by. And my sister still to this day is jealous about the fact that I won the dance contest and she did not. So little sibling rivalry there. Anyway, I'm so delighted. I was so delighted when Dean Clark asked me to be the speaker for homecoming. I just think that, you know, where else at Berkeley do you get this many people in the room at homecoming to listen to a science talk? And, just remember, I think this is the first homecoming in person in 3 years. So all of you, you should look to the person to your right and your left and just say, go Bears, right? This is fantastic. Okay. All right. Okay. Right, and so here is, right, the requisite homecoming slide. Okay, so I was told in very certain terms, that one of the expectations for this talk is for me to describe my path to Berkeley. And I have to say, I'm really not comfortable I like to talk about science. I'm just a total nerd, dance contest notwithstanding. And so it's really only going to be one slide, but there is a funny story that's related to the dance contest anecdote. All right, so you might be able to tell from my accent that I did not grow up in California. Okay, true. I grew up in Rego Park, which is a part of Queens. How many of you know where Queens is? Awesome, awesome. Okay, so I grew up in Rego Park, and I went to public schools in Rego Park and went to college a little early because my mother was aggressive with the kindergarten teachers. Spent 4 years at another public university, a flagship university, not of California, but of New York, at the State University of New York at Albany. After college, where I majored in chemistry, of course, I went to Columbia University for graduate school, met some of the best friends of my life there, Spent 4 years in Ronald Breslau's lab discovering the field of chemical biology, which I'm going to talk to you a little bit about today. And then had a brief foray to California, because I spent about 2 years at Caltech as a postdoctoral fellow in the lab of Peter Dervin, who some of you may know, some of you may recognize that name. And then, although I told my mother when I looked for jobs at Caltech that I was never going to leave California because I loved it so much, this is true. She reminds me of this. You know, the cards were not on the table for a job in California, and I ended up spending 30 years, 31 years to be exact, in beautiful New Haven, Connecticut, at Yale University. And this image that you see there was painted by one of my former colleagues, Harry Wasserman, an organic chemist who spent his entire career at Yale. He focused on synthetic organic chemistry. He pioneered many novel reactions, including those of tricarbonyls, if that means anything to you. And he was a great and dear friend. And he was also a phenomenal artist. And this is a painting, a watercolor painting that he made of Sterling Chemistry Lab. But, you know, 32 years is a long time, or 30 years is a long time. And in 2018, I was so delighted to be recruited to come back to California to UC Berkeley. And I just have to say that there is nothing, I have nothing but positive things to say about this move. I love the fact that when I walk around campus, I hear students talking about science and their intellectual pursuits. I love the fact that collaborations here are part of the natural culture and are not forced. I love my colleagues, and I have to say that the caliber of the undergraduates here is unlike anything I have experienced at any university. So kudos to you for choosing Berkeley. Like, I just have to say that the education that the undergraduates and graduate students get here, as someone who has not spent their entire year at Berkeley, it is second to none. So it's just such an honor for me to be able to teach such remarkable students. And I hope to be able to share some of our recent work with you, and at a level that everyone can appreciate, including the puppy that's over there. We're hoping that we don't get a bark in the middle of it. We'll see, we'll see. I almost brought my dog just because I knew that Mindy was going to mention. I have a pair of Bouviers, and one of them is 100 pounds, and that probably would have been a little too big for the room. Anyway, so when I came to UC Berkeley in 2019, I brought a collection of graduate students with me from Yale. The group has now grown considerably. This was our first group party after in the lull of the pandemic. So we've now had a second group party, and the lab has expanded since then. And now, I think in about a year, it will be completely Berkeley students. And I'm very delighted to share my enthusiasm for science and my love for Berkeley with these students on the 4th floor of Tan Hall. Okay, so one of the other things that I brought to Berkeley when I moved from Yale was a center, an NSF center called the Center for Genetically Encoded Materials. And so this is NSF has a fantastic program called Centers for Chemical Innovation, where they give groups of scientists a lot of money, $20 million, to solve problems that are so difficult that they border on what would be imagined as science fiction. And I was very fortunate to lead a team that managed to secure one of these NSF grants. And that's one of the 3 areas of research that we work on in our labs in Tan Hall. And this is a very collaborative project. It involves many labs at Berkeley, a few labs other places, but many labs at Berkeley. And I'm going to try my best to share some of the discoveries that we have made since we've arrived at Berkeley, mostly in collaboration with other Berkeley labs, with you in this talk today. Okay, so I think the best way to introduce what the Center for Genetically Encoded Materials is trying to do is to really point out something that everyone knows but doesn't really talk about, which is that in the world today, there are really 2 types of polymers. There are biopolymers, which are typically made by organisms, and then there are plastics, okay? And these 2 families of molecules have some things in common, but many things are different. And so I think the best way to describe the challenge that the Center for Genetically Encoded Materials is trying to address is to point out what the differences are between biopolymers and plastics and the advantages that each family of molecules has relative to the other. Okay, so I'm actually going to move now so that I can point to the slide. Okay, so one type of polymer that exists in the world today are biopolymers. And what is unique about a biopolymer is that although, like all polymers, it consists of many individual monomers that are linked in a chain, in a biopolymer, those monomers are different from one another. So there are three types of biopolymers that exist. There are proteins, there is DNA, and then there's RNA. We're not going to talk about oligosaccharides. That can be an argument for after the talk. Okay, but what is unique about biopolymers is that whether you're talking about proteins or DNA or RNA, their overall chemical diversity is actually quite low, right? So for a protein, proteins are composed of linear arrays of monomers that are called alpha amino acids. And in most organisms, there are really only 20, maybe 21 in some organisms, but really only 20 that are inserted into polymers at the level of the ribosome. Okay, for DNA and RNA, the diversity is even lower. It's only about 4 monomers. And so 4 different colors of different monomers along this string, and 20 in the case of proteins. So the other unique feature of biopolymers is that in all cases, their synthesis is templated. And so the information that tells an organism what the sequence of individual monomers should be for a given protein or DNA or RNA is templated by another polymer. And in proteins, the individual monomers are linked by amide bonds. And of course, in DNA and RNA, they're linked by phosphodiester bonds. But no matter, the sequence is 100% encoded. Okay, so there's information that tells what sequence the protein should have. And for proteins, one of the elements that fascinated me 30 years ago and still fascinates me today is that from this defined sequence of only 20 different monomers arises what is often a unique structure, not always, but often, but always a sophisticated function, and function that is sophisticated enough to keep each and every organism that exists on Earth alive. Okay, so the advantages that biopolymers have, specifically proteins, is that they have a precisely defined sequence and length and a unique structure. The disadvantage is that their chemistry is limited by the number of monomers that can be added into the chain. For natural proteins, the limit is about 20. But even with that, right, these are all very recently published structures of proteins so that you can see the beauty that these molecules, the structural beauty of these molecules. And so whether you're thinking of, the Cas9 gene editing machinery as characterized biochemically by the Doudna lab, and then biochemically and structurally by the Doudna and Nogales labs at UC Berkeley, or you're talking about the ribosome, which we will be talking a lot about today, as characterized in beautiful atomic detail by Jamie Cates' lab. I think Zoe Watson is in the audience, and she solved this structure. Whether you're talking about the active state of soluble guanylate cyclase, which is involved in NO signaling, as solved by the Marletta and Hurley labs over here, or even this really novel radical halogenase that replaces hydrogens in substrates with chlorine, okay, as engineered by Michelle Chang's lab. So these are all beautiful structures that have come from UC Berkeley lab that really emphasize the sophisticated function that can emerge from even a natural biopolymer that's composed of 20 alpha amino acid monomers. Okay, so now let's go to plastics. So plastics have different advantages. Okay, so in contrast to proteins, they have very high chemical diversity, right? In fact, although most polymers have the same monomer at every position along the chain, as illustrated by this figure, in contrast to proteins, where the individual monomers have a defined sequence, right? In most plastics, all the monomers are the same. They're all linked to each other, but the diversity of chemistry is unlimited. So the monomers are limited only by your imagination. The bonds that link these monomers together can vary enormously, esters and amides and carbon-carbon bonds. The difference is that the sequence of a plastic is in general not templated, okay? And the order of monomers, even if you have two monomers that have the potential of forming a polymer in the mix, the relative order in the chain is really dictated simply by relative rates. Okay, so it's a kinetic effect. There's no templating that says, I want A, B, C, D in that order in the polymer. But still, the functions are amazing. I mean, where would we be? I'm not sure if this is a good question. Where would we be without polystyrene? Okay, where would we be without crazy glue? Okay, or, nylon, or, Polartex. These are all materials that are around us everywhere, that emerge from the fact that you can diversify the chemistry of polymerization, right, to generate materials that have such diverse processes. And so the question that CGEM is asking is, so the advantage of plastics is that they're really almost unlimited monomers and linkages. But the disadvantage is that they have a poorly defined sequence and length, because no one has yet figured out how to make a plastic that has different monomers in a uniquely defined array, like the uniquely defined array of monomers that are found in proteins. That's the challenge that CGEM is trying to solve. We're trying to use the translational machinery, the ribosome, which makes proteins in all organisms on Earth, to make materials that don't look like proteins anymore and begin to look like sequence-defined plastics. Because we are confident that from those sequence-defined materials will emerge properties that are unlike anything that can emerge from a natural protein. And in fact, there's already some evidence of that. So this molecule shown here, it may look to you like a protein, but it's actually not, right? So that is an octamer, 8 copies of a beta-peptide. And so these are amino acids that have a different backbone than the alpha amino acids that are found in the proteins in all of your cells. And when incubated at reasonable concentrations, this one sequence of a beta peptide self-assembles into that structure, which looks like a protein, but it's actually closer to an aggregate of nylon than it is to to a protein found in nature. That superstructure, which Doug Daniels prepared in my lab, more than 10 years ago, that beta peptide bundle, as we call it, has a melting temperature of above 95 degrees, despite the fact that it's simply an octameric assembly of 8 helical beta peptides. And so it's clear that unique properties can emerge from materials that have backbones other than the alpha amino acid backbone found in proteins. We simply need a way to use something other than solid phase synthesis to make those molecules to scale. So that's the challenge that CGEM is trying to solve. Yeah, so the question is, what if chemical polymers could possess defined sequences? And what if we could make a protein where a region of its sequence is actually not an alpha amino acid array, but instead an array of a family of natural polymers that are known as natural products? And so those are the stories that I will try to tell you about today. Okay, so the way that CGEM is approaching this problem is from 2 very different directions that I think most people could easily appreciate as described as bottom up and top down. Okay, so bottom up is, what we're asking the ribosome to do is, rather than make a bond between an amino acid in one region of the ribosome active site that has a nitrogen nucleophile and an ester in the other active site of the ribosome, rather than making an amide bond between an amino group and an ester, okay, we simply want to use the ribosome active site to make bonds between things that are not alpha amino acids. Okay, and so I'll tell you 2 stories today that describe steps that are needed to achieve that goal. The second way that CGEM is approaching this problem is, rather than asking the ribosome to make novel bonds itself, we say, okay, let's let the ribosome make what it wants. And then asking, can we use chemistry? Can we use enzymes or catalysts or just chemicals to take that translated protein and convert it into something that now has a backbone that isn't an alpha amino acid backbone? And so I'll tell you a short story about how we've been able to achieve that at UC Berkeley. Okay, so I think the first thing I want to describe is some of you may be thinking, well, why is this so hard? Like, why is it so hard? And part of it is hard because, of course, the ribosome has evolved for billions of years to make one bond, right? It's a bond between two alpha amino acid monomers. And you can imagine that organisms have evolved multiple strategies to make sure that it doesn't form bonds between other things. And so so the truth is that even today, in 2022, the number of examples in which a ribosome has been used to make a bond that is something other than a new amide bond between two alpha amino acid monomers, you can count it on one hand, okay? All right, so the first example was actually carried out here at Berkeley. It wasn't in our lab, we were at Yale at the time, it was in Peter Schultz's lab. And so what Peter Schultz's lab did here at Berkeley in 2008 is to show that you could make very simple changes in the backbone of a protein rather than using monomers that are amino acids. He simply used monomers that were hydroxy acids. And so this would be tyrosine, the natural amino acid tyrosine, if this OH group was instead an NH2 group. And he demonstrated in 2008 that the wild-type E. coli ribosome in cells was perfectly capable of making a bond between that nucleophile, OH, and the ester that exists in the P site to form an ester linkage in a protein, right? Breakthrough result. It was done a little differently by another lab a year later, and that really is where the field stood until 2016 or so, or 2016. And so what our lab did back in 2016 is to say, okay, we know from all of the work that has been done in Peter Schultz's lab and many other labs by that point, that the ribosome is actually pretty tolerant of an alpha amino acid backbone if there are even wild changes in the identity of the side chain. What we wanted to know is whether or not the ribosome could introduce a monomer into a protein that had a different backbone with a completely different geometry. And so Wes Robertson in the lab used a pretty clever strategy to identify mutant ribosomes that had RNA base changes within the peptidyltransferase center, which is the active site of the ribosome where the amide bond forming events occur. He identified mutant ribosomes that were able to insert a single monomer of a beta amino acid into a protein in vivo. And so this is an example of a protein dihydrofolate reductase that at position 132 doesn't have phenylalanine, it actually has beta phenylalanine. And so this was the first example, and in fact, for many years, the only example, in which a protein had been made in a cell that had anything other than an alpha amino acid backbone in its sequence. Okay, the second example of, but let me just point out that this required an evolved unnatural ribosome. So a second example was published a few years later. So this is the work of Sid Hecht, who's at the University of Arizona. And what he showed is that you could insert this, kind of interesting fluorescent monomer into a protein, again using an engineered ribosome, to make bacteria that stain blue. Okay, but these are the three examples, or 3 or 4 examples. That's it. And so the first question you may ask is, well, why is it so hard? Like, why is it so difficult for the ribosome to introduce other monomers into a protein in vivo? And the answer is very simple. The problem is not the ribosome. The problem are the enzymes that are used to load monomers on tRNAs, which are the ribosome substrates. Okay, so today, so what was known is that it turns out that if you look at Jamie Cate's structure, or Zoe Watson's structure, if you look at the active site of the ribosome, where all the bond-forming reactions occur, there's room for more. And in fact, in vitro, where you could, not in vivo, but in an Eppendorf tube, where you can load up on the concentrations of tRNA, you can load up on magnesium, you can crowd all of the reagents. It turns out that many different monomers can be added by the ribosome. The problem in vivo instead, is that there are no enzymes available that are able to load monomers that are not alpha amino acids on tRNAs. Okay, so then we'll get to the first story here. And so the first story that I'll tell you about, work done completely here at UC Berkeley, right, is our ability to identify enzymes that actually incorporate in vivo non-alpha amino acids onto tRNAs and then into proteins. Oh, good. Well, he knows what to clap at. It's a big deal. All right, that's going to be the longest story. And I have to say that we would not have gotten very far in that project were it not for a fantastic collaboration with Christine Guy, who's in John Currien's lab, who helped us solve a crystal structure, even though it's been literally 20 years since I solved the crystal structure. So that was really important. Then there'll be two shorter stories. The first one focuses not on the enzymes that load amino acids on or monomers onto tRNAs, but instead focuses on that ribosome PTC. Because I think what you'll appreciate after the first section of this talk is that it's actually really hard to identify those enzymes. It is not a trivial exercise. And so then you can't just try to identify them willy-nilly. Okay, you need some guidance on which monomers are going to be the best substrates for reaction in the ribosome PTC. And that's a fantastic second story I'll tell you about. Work that was led by Zoe Watson in Jamie Cate's lab in collaboration with our lab, as well as a collaboration with Schrödinger, which is a fantastic computational chemistry company that is both in New York and California. And then finally, so both of those short presentations will be about the bottom-up approach, where we're trying to form new bonds in the ribosome active site. And then we'll have a very quick foray into some very recent results from our lab, where we've been able to execute the top-down approach, where we're actually able to use kind of cool biosynthetic enzymes to take a protein, a fully folded protein, and in a site-specific way, introduce an aromatic ring, a thiazolene, in the backbone of a protein for the very first time. Okay, so those are the three stories. So the first two are bottom-up. The first one will be the longest, and the second two will be very short. And there'll be a second bottom-up project and then a top-down project. Okay, so just because I appreciate that it's been, perhaps not everyone is remembering the biochemistry of translation, okay? I've been talking about these enzymes that take monomers and add them onto tRNAs, but that's a chemical reaction. It's an acylation reaction, right? And it's carried out in cells by enzymes that are called aminoacyl tRNA synthetases. So just a little bit of the chemistry of aminoacyl tRNA synthetases. So as I said, these are enzymes that acylate tRNA, a hydroxyl group, a specific hydroxyl group, on tRNA with alpha amino acids. The reaction involves two separate chemical steps, which we'll talk about in just a moment. It's important to know that in most organisms, there's one enzyme that is responsible for each natural alpha amino acid. And so for tyrosine, there's one aminoacyl tRNA synthetase that loads tyrosine onto a specific tyrosyl tRNA. And there's one enzyme for leucine, and there's one enzyme for phenylalanine. And so in most organisms, there are 20 different aminoacyl tRNA synthetases that each have the job of generating the substrate that is needed to insert that amino acid into a protein. Okay, and the other thing that's important for you to know is that, the tree of life is big, right? The tree of life is big, and the enzymes that exist in archaebacteria will operate in E. coli, in proteobacteria. But in some sense, they're ignorant of the fact that all of those E. coli enzymes and tRNAs exist. That's called orthogonality. And so that means that if you install a new activity in an archaeal aminoacyl tRNA synthetase, that activity will be retained in E. coli, and there'll be no crosstalk with any of the E. coli enzymes or natural amino acids. And so that's a good thing. And so this is the chemistry of aminoacyl tRNA synthetases. It's a two-step reaction. Here is an amino acid, an alpha amino acid, which has a amino group and a carboxyl group, and this is the state that they exist in at pH 7. And the first step of this reaction is nucleophilic attack of that carboxylate on the alpha phosphate of ATP to generate what's called an aminoacyl adenylate. And those of you who remember organic chemistry or remember that carboxylic acids are terrible at acyl transfer reactions because OH is a crappy leaving group. But there are ways to make OH into a much better leaving group, and linking it to ATP is a fantastic way to make a hydroxyl group into a fantastic leaving group. And in fact, in the second step of an aminoacyl tRNA synthetase reaction, a nucleophile on tRNA, which is specifically the 3' hydroxyl group on the 3' end of tRNA, is bound by the enzyme in such a way that it can then attack that phosphate, attack the carbonyl carrying the phosphate, to move this amino acid group onto the hydroxyl group to generate the acylated tRNA. Right, which I've drawn in the wrong way. Oh my goodness, there's a mistake. Okay, we're going to take this slide off. I can't believe that. Okay, no one noticed that. Anyways, you could tell that I was doing this at like 2 in the morning. Okay, wow, I can't believe that. That's a first. Okay, so as I mentioned, right, archaeal enzymes and bacterial enzymes don't talk to one another, which is fantastic. And the other fantastic thing is that not all archaeal enzymes are the same. And in fact, because if they were the same, I would not be able to share any of these cool results with you today. And so what I show you here is a very strict close-up of the active sites of 2 archaeal aminoacyl tRNA synthetases that are extremely well studied and often used for what we call genetic code expansion, which is simply to insert unnatural alpha amino acids into proteins. Okay, and so the enzyme shown on the left is from an organism known as M. ganeshi, and the enzyme shown on the right is from an organism known as M. mazie. Okay, and you know they look different but the same. Let me just point out what's important. So if you look at the enzyme active site on the left, what's shown in orange is the substrate. So this is tyrosine. This is the natural substrate for this enzyme. And this blue bar here, that's the amino group of the substrate. Okay, and what you can see here is that that amino group is linked through 3 dashed yellow lines to 3 side chains on the protein. And those yellow lines are hydrogen bonds. And so what this says is that the nitrogen on that substrate is hydrogen bonded directly to 3 side chains on the protein. Okay, it's held there very specifically, and it's held in such a way that the carboxylate on the amino acid can attack ATP, which is bound down here. Okay, let's just, before I pass judgment on that active site, let's look at the other active site. So this is the enzyme from M. mazie. Okay, so now the substrate is a little different. It's pyrrolysine, which is an extra amino acid that exists and is used in some archaeal enzymes. And so again, in orange, we have the substrate for the enzyme. And again, here is the amino group of this alpha amino acid. And so what you see here is that, again, there are all these yellow lines, but there's also this red dot right in the center. That red dot is a water molecule. And so what you should be able to get out of this picture is that the active site of this enzyme from the M. mazie enzyme, right, that nitrogen is not directly hydrogen bonded to anything on the enzyme, right? Those interactions are all mediated by this very well-placed water molecule. So the water molecule is donating and accepting hydrogen bonds to different side chains on the enzyme, but the amino group is simply bound to the water molecule. So what that suggested to us is that, one, if the water molecule was not there, there would be a lot of space there for substituents on that nitrogen or backbones of a monomer that differed from an alpha amino acid backbone. And in fact, it was already known, right, that in the enzyme shown on the right, that the amino group of a natural amino acid could be replaced by hydrogen or by an OH, by small changes. And so the question that we asked is whether or not we could replace that NH2 with things that were longer, that would allow us to insert monomers into proteins that had alternative backbones using an enzyme. Okay, and the answer is yes. Okay, and the answer is yes in actually a way that is even more interesting and exciting than just making proteins with novel backbones, because it provided us with substrates that we think will allow us to make proteins that have carbon-carbon bonds instead of carbon-nitrogen bonds. Okay, so these are all the substrates that we have found for these related enzymes. And so some of them have OHs instead of NH2, some of them have thiols instead of NH2, some of them have, formamides instead of NH2, but the really interesting ones have these functional groups, where instead of an NH2 attached to the backbone, you have a carboxylic acid. Okay, and you're probably thinking, so just FYI, so this is the first and only example of a synthetase that can acylate tRNA with alpha-thiol acid, which allows us to make proteins with thioester backbones. The first and only example of a synthetase that acylates tRNA with a malonate. Okay, and the question is, why are malonates so interesting? Because everyone thinks of malonates as being electrophiles, not nucleophiles. But I just want to show you the chemistry in two active sites here. So on the left is shown the chemistry that happens naturally in the active site of a ribosome. So you have an amino group, it attacks an ester, and you make a new amide bond. On the left, on the right, is shown the active site of a different family of biosynthetic proteins that make natural products known as polyketides. Okay, polyketides like tetracycline, erythromycin, all sorts of really fantastic natural products. And the fundamental chemistry that makes those natural product polymers is the reaction between an enolate that is formed by decarboxylation of a malonate half ester and attack of that on a thioester. And the fact that we can now generate a tRNA that carries this latent nucleophile is the first step in using the ribosome to make the carbon-carbon bonds that are found in polyketide natural products, as opposed to the carbon-nitrogen bonds that are found in proteins. And that's one of the things that the lab is currently working on very actively. Okay, we were really interested in how these malonates bound to the enzyme, and so Cameron Swenson, fantastic graduate student, solved the structure, and he and Riley Frick characterized all the biochemistry that I described to you. And the structure itself is, just unbelievably cool. So here is the substrate, the side chain of the enzyme, and what you can see is here's the carboxylate that actually reacts with ATP. And what you can see is that the other carboxylate, the malonate, instead of the water molecule is completely gone. But now what is really cool is that now that carboxylate is engaging with the same backbone NHs that the water molecule was engaging with in the structure I showed you previously. So by taking out the water molecule, we basically reversed the preference for being a hydrogen bond donor or acceptor and allowed us to generate an enzyme that can actually bind a malonate, use it to acylate tRNA, and we're now working very hard to try to use that tRNA to make carbon-carbon bonds on the ribosome. Okay, so that's the longest story. Two very short stories to finish up. So it's clear that making these synthetases is not easy. Okay, and so, but what synthetases do you want? Like, how are you going to decide what monomers are going to react the fastest within the active site of a ribosome, even a wild-type ribosome? And so for that, we turn to calculations. And so, in a collaboration, as I mentioned, with Jamie Cates' lab here at Berkeley, and also with Schrodinger, a fantastic computational chemistry company. Okay. Okay, and so the question we wanted to ask is it possible to predict the reactivity of something other than an alpha amino acid in the active site of the wild-type E. coli ribosome? And so in some sense, we began with just some facts. Like, it is well known that nucleophilic attack on carbonyls prefer defined angles. So how many of you remember what the Berge-Dunitz angle is? No one. Okay, great. Okay, so it turns out, well, so I'll tell you what the Berge-Dunitz angle is. So it turns out that both calculations and experiment have demonstrated that when you have a carbonyl, okay, so the carbonyl has a flat pi system, okay, that the nucleophile prefers to attack from an angle between 90 and 110 degrees. Okay, and that's what this angle is. It's called the Bürgi-Dunitz angle. And that angle exists because, of course, you're having a nucleophile with a highest occupied molecular orbital, and those electrons are adding into the pi star orbital of the carbonyl. And that has specific geometric preferences. And so we knew that that angle was important. There's actually a second angle that matters, and that's an angle that actually comes from work from UC Berkeley. It's a Clayton-Hethcock angle. It's called the Lodge-Hethcock angle, Hethcock-Lodge angle. And it turns out that's also a preference if the carbonyl is basically if you're looking straight at the carbonyl, it's not just the angle relative to that plane, but sometimes it's one to one side or the other side, depending on the size of the two substituents on the carbonyl. So that's called the Flippen-Lodge angle. And so we knew that these angles were important. And so we hypothesized, right, that the monomers that would react the best would be ones that would occupy free energy minima where the nucleophile was close enough to the carbonyl and then occupied these Bürgi-Dunitz the preferred Bürgi-Dunitz and Flippen-Lodge angles. And so the first experiment is to identify a computational workflow that would give us these free energy minima. And so this is just an example of a plot that shows the Bürgi-Dunitz angle versus the distance between the nucleophile, the amino group, and the carbonyl ester. And what you're looking for in these plots are the dark blue portions, because those are the minima in the curve. So you can think of that plot as being a big contour, where the minima, the lowest energy, is where you see the darkest blue. And then we started with a test set. Okay, so there had been a lot of studies on the in vitro translation of monomers that have very diverse structures. And so we simply asked whether or not we could devise a computational workflow that would reproduce the relative rates at which these monomers are incorporated by wild-type ribosomes. And I have to say that the data is just so beautiful, because what you can see for 3 different families of monomers, whether they're aromatic rings or cyclic beta amino acids or linear beta amino acids, in all cases, what we observe using metadynamics is that these free energy minima plots trend perfectly in agreement with the relative rates of translation, telling us that this method can be used very quickly for us to investigate large numbers of monomers and identify the ones that are most likely to translate the best were we to have an amino-acyl tRNA synthetase. So for us, it helps us understand the requirements for this reaction, but it also helps prioritize our research program so that we know which amino acid tRNA synthetase enzymes to evolve and design. Okay, that's the second short story. The final short story is now the top-down approach. And this is really very quick. Okay, and so this is where we're basically taking a protein and asking, can we generate an enzyme that can take the backbone of a protein and basically remove it and edit in a backbone that is completely different than what you see in natural proteins. And so everyone knows about gene editing. This is an example of protein editing. And this is work carried out by Josh Walker, a postdoc in the lab. And so I think if you start talking about protein editing, you have to think about this natural family of enzymes that make natural product known as RIPs. So RIP stands for ribosomally synthesized post-translationally processed peptide. It's a handful. That's why we say RIP instead. And it turns out that nature does this all the time. So there are lots of natural products that are really important medicinally, pharmacologically, for research, right, that have crazy backbones, like really crazy backbones. So these natural products known as polythionamides, I mean, you may look at this, you say, that looks like a protein. And it does, except more than half of the alpha amino acids in this protein have their stereochemistry switched. So rather than being L, they're D, which leads to unique pharmacological properties for that molecule. And so these are enzymes, and so there are enzymes that take an L amino acid and turn it into a D amino acid in a sequence-specific way. And there are other enzymes in the context of that natural product. There are other enzymes that have a substrate in which they literally rips out an element of the backbone and ligates it together to generate a beta amino acid. There are others that do crazy things like Diels-Alder reactions to generate pyridine rings in the middle of natural products. And finally, there are enzymes that also form these thiazolene linkages within sequences of natural products. Now, for these RIPs, the enzymes are very specialized. And so there's a RIP enzyme that acts on a unique substrate to make a unique natural product. What we wanted to know is, could we repurpose any of these enzymes so that now, rather than acting on the natural product substrate that they evolved to act on, they acted on anything. Anything that we give them that has the right, core sequence, can they take that core sequence in a fully folded protein and add in the chemistry that they're designed to add in? And the answer is yes. Okay, so it's getting a little, I'm just going to go quickly. Okay, and so the experiment that we did, so we're using a family of enzymes that carries out a reaction known as a cyclodehydratase reaction. So a sulfur side chain adds into a carbonyl, you lose phosphoric acid, and you generate a thiazolene. And so the question we asked is, could we install this thiazolene in a backbone, in a normal backbone? And so Josh made a series of analogs of a protein known as ROP, which has 2 alpha helices and then a loop. And he inserted the sequence that an enzyme known as MICD recognizes, so that the reactive cysteine in the enzyme is that orange ball. And then he asked whether or not a really unique constitutively activated form of MICD could take that cysteine and convert it into a thiazolene. And in fact, that's exactly what we were able to observe, that in ROP and in other protein substrates, that all you really need is a relatively unfolded, a region of the protein that is not involved in alpha helical or beta sheet formation. And that MICD, this unique activated form of MICD, will convert that cysteine into a thiazolene. And if we add in a second enzyme that oxidizes that thiazolene into a thiazole, that we could generate a protein, let's actually go back, that rather than having a cysteine side chain, has a side chain that now has a fully aromatic ring within the backbone. And you could imagine that if we could link these together, that there might be unique energy transduction properties. And if we could link them together in a different way, then we would have a way to take a protein and install in its backbone something like looks like the natural products that I described to you on the first slide that introduced this topic. And those are some of the experiments that we're now working on in the lab. Okay, so those are the 3 stories that I'm hoping I shared with you. I was able to communicate with you today. So we have, in these 3 all Berkeley stories, we were able to generate some new activities of unique archaeal amino acyl tRNA pairs that allow you to introduce novel substrates into proteins in vitro and in vivo. I've shared with you a computational approach that helps us identify which monomers we should work on, which is fantastic. And then I've described this completely new strategy to take the backbone of a natural protein and convert it into something that now looks more like a plastic. It's not actually a plastic yet, but it looks much more like a plastic than it did before. And so I'd like to thank you all for coming to my talk. I'm just amazed at how many people are here. And this is the group, and these are the agencies that fund our research. I'm really incredibly grateful to all the members of CGEM, to Jamie Kate and his lab and Zoe Watson, to John Kurian and Christine Guy for helping us with the crystal structure. And once again, I'm just so grateful and so honored to be able to present this work to you today. I'd be happy to answer any questions that you have. Oh, and we have a mic for the questions. Just please don't ask about dancing. Okay, awesome.
Actually, for a non-chemistry person, here I want to take away for your lecture. Can you give me like 5 keywords? And based on the order you think important.
Yes. 5 keywords. Wow. Sequence-defined polymer. Shelf-stable protein. Antibody-drug conjugate. Synthetic biology. And great Berkeley students.
In the beginning of the talk, you mentioned briefly that this chemistry
could you highlight some of those probably projections?
Oh yeah, I mean, I think the first example. So what if, imagine what if biologic drugs, antibodies, insulin, COVID vaccines, what if they would not need cold chain storage? What if? What if they could just be transported at room temperature? They wouldn't need a refrigerator in parts of the world where refrigeration is difficult. Imagine, right? And so the reason we're so interested in the top-down approach is it's clear that installing these, so I didn't actually talk about it in detail, but those thiazolines and thiazoles, because they're full of sp2 hybridized atoms, they constrain the backbone in very unique ways. And so the real home run for that portion of the effort is to use them to replace loops in proteins so that the proteins once folded do not unfold anymore. And that is definitely science fiction, but I think it's an element of science fiction that is actually within reach. So I think that's the best example.
So on that note, so then if you have a non-natural protein used as a therapeutic or vaccine, can you comment on the toxicity?
Oh yeah.
What the body is going to do when they see that?
That's also, in some sense, you worry, okay? Because of course, if you have a sequence of a protein that has unnatural backbones and it's presented by MHCs, there are immune reactions. But the beauty of the monomers that we're trying to insert is that they are not proteolyzable. So it turns out that beta amino acid linkages are not substrates for most proteases. And of course, if proteases don't degrade your protein, you don't generate the short peptides that can be presented by MHC molecules. And so there's a reason to believe that if a protein can't unfold, it also can't be substrate. It certainly can't be a substrate for the proteasome. And it won't be a substrate for many other proteases that demand an extended sequence in their active site and multiple amino acid dependent interactions along that chain in order to generate the structure that as acted upon by the enzyme. And in fact, it's well known that in cells, peptides that contain beta amino acid linkages are not proteolyzed. So I think that the toxicity is a multivalent, certainly there are elements of toxicity that have nothing to do with immune recognition, right? And so there are issues about how the material would be metabolized. There are issues with regards to how it would be trafficked in an organism. Those are all things that we can't approach until we have materials that we can inject into animals and people. But it's an important approach, but I think that what we're doing has advantages because the monomers that we're inserting will make the material less proteolyzable and hence more stable in a cell than they would be otherwise. I think she was first. Sorry.
Can you elaborate on why you said polysaccharides you didn't use polymers?
Yeah, no, they are polymers, and they're very complex, and they have really unique structures. And I think that they're just not templated.
You mentioned earlier that the insertion of those valid groups in that first story you were talking about was related to the PKS system. It sounds like you're repurposing the PKS chemistry into ribosome. I'm wondering about the current state of the field in terms of PKS molecule engineering to make counterfunctional.
People have been working on that for at least 20 years. It's been very difficult. It turns out that the machines that make polyketides in cells are very complex. And so it's a machine, right? It's an ordered machine where every individual reaction is carried out by a different protein module, and they all are engaged through these linker regions that have been very difficult to re-engineer. And so although there has definitely been some progress in the use of PKS engineering to make unnatural natural products, unquestionably, it's not templated in the same way that an mRNA sequence is. And so our approach is different. It's a little simpler, right? Because the ribosome is one thing. We're engineering one thing as opposed to many enzymes in a pathway. But our idea is then you simply use the mRNA to dictate exactly the order of individual monomers. And so I would say the two approaches are very complementary and have different strengths and weaknesses. Yep. They can exist. So the experiment that I described that was published in 2016, in that case, we grew E. coli cells that actually had 2 types of ribosomes in them. They had the wild-type ribosomes that the cell was using to keep itself alive. And then it had this other family of ribosomes that were the only ones in the cell that were able to incorporate that one monomer of a beta amino acid. And they coexist. Of course, they compete for ribosomal proteins, and they compete for mRNA. Others have generated ribosomes that are unique in that they accept only certain mRNA sequences as substrates. And so that can allow you to generate a situation where you have 2 ribosomes in the cell that act on 2 families of mRNA messages. And so those ribosomes are said to be orthogonal because they prefer mRNA sequences with a unique upstream sequence that is different than the upstream sequence that is recognized by wild-type ribosomes. So the answer is yes, they can coexist. That's not our work. Hi. Ooh, well, CGEM has a website. Okay, it's online. You can easily find it. It's GemNet, and we have some tutorials on that site that can give you a background on our work and how it fits into the larger context of research on translation. It also tells you about, CGEM actually is more than just research. It involves all sorts of training and educational and broader impact projects and ideas and, things that we're doing, and you can learn about that there as well. Okay, so thank you. Thank you. Go Bears.